Okay, so what happens when there's errors in this process of taking DNA to RNA to proteins? What happens if there's errors? And that what happens is a genetic mutation. So mutation is a change in the DNA nucleotide sequence. So actually changing the sequence of your DNA is what the mutation is. They may alter the sequence of amino acids, so it might change what that protein is. It might affect the structure and function of the protein within that cell because you changed the sequence, you changed the primary sequence of amino acids. So mutations, how they happen is through x-rays. So we talked a little earlier about Rosalind Franklin. So she worked a lot with x-rays, but didn't realize that it was going to cause mutations in her body. And so she died really early from cancer because of it. And then mutations also could be overexposure to sun. So that's how you get skin cancer, is from extra UV light or excess UV light, not wearing sunscreen, overexposes you to sun and mutates your DNA. Additionally, you can have chemicals called mutagens. So there's specific chemicals that actually will change your DNA, which are very dangerous, and you have to be really careful when you're working with them. And then some viruses can also cause genetic mutations. Okay, so mutations in a somatic cell, so not a reproductive cell, so like your skin cells or your organs or something like that. It's limited to that cell and its daughter cells. And so the cell replicates, forms one cell, and there's a mutation. Then if it replicates a lot, or if this does cause uncontrolled growth, that's what cancer is. So when you have a genetic mutation in a cell and it just repeats over and over and over, typically cancer, whatever that genetic mutation is, it causes the cell to just keep replicating over and over and over and over really fast. And that's what you get tumors and things from. However, if the mutation is in a germ cell, so an egg or a sperm cell, that is the DNA responsible for making that next human, right? So the individual will contain the same genetic change. So their entire being will have that genetic mutation. So types of mutations, there's point mutations or substitute mutations. They're called similar things. This is where you just take one nucleotide that's incorrectly put together and it switches. So for example, I wrote this really small, sorry guys. For example, you could have CCG changed to TCG. So it then would code for, it doesn't necessarily have to, but it could code for a different amino acid, but it would just be one, it wouldn't be a whole chain of differences. So if it's a silent mutation, then there's no change in amino acid sequence. So there are multiple codons for the same protein in some cases. And so there could be a mutation that just doesn't cause anything to change. There could be a missense mutation, meaning that a nucleotide changes codes for a new amino acid. So like up here, what I drew, if you change this CCG to TCG, you have glycine originally, but now it's serine. So this would be a missense mutation, meaning it's a mistake that changes the amino acid. A nonsense mutation is a codon that it changes the codon to stop. And so it just stops the protein altogether from being, or it stops it at that point. There's also something called, so this is like a single, like one nucleotide changes. There's also something called a frame shift mutation. So the base A nucleotide is inserted or deleted. So everything shifts one, either shifts one extra or one away. And this usually results in a completely different amino acid sequence. You can imagine if you just move something over, everything's going to be completely different in terms of those three pairs. So you could have a deletion mutation where one's deleted, or an insertion where it's one's inserted. So frame shift mutations are pretty rare, but they're typically like very destructive towards amino acid sequences. So quickly, the effects of mutations, so the change in amino acid could change the protein structure. So it changes the primary structure, right? But that could also change the secondary, tertiary, quaternary structure of that protein. So for example, if it's an enzyme it might not bind to its substrate anymore. It might bind to the substrate, but not react anymore. So things like critical syntheses wouldn't occur, so you couldn't make molecules that you needed for that cell to function. You could also accumulate toxic molecules, so it could be fatal. But what I mean by fatal is not like kill your whole body, it just kills that cell. So that cell would die. New cell would take its place in most cases. And then there's some genetic diseases that are formed typically from those germ cells having mutations. One is called phenylketonuria, or PKU. This is where your DNA cannot directly synthesize the enzyme phenylalanine hydroxylase, which is required for the synthesis of phenylalanine to tyrosine. So tyrosine is something that our body makes. It just takes phenylalanine and adds that OH here. But people with PKU do not have in their DNA the code to make that enzyme. And so it's really dangerous for them to get excess of phenylalanine, and it could cause a lot of problems in their system without having the tyrosine there. So things, for example, like this molecule here I'm blanking on the name right now. It's one of the fake sugars that they use. But this molecule here tastes sweet to us, but it's an additive in some sodas. And people who have PKU have to be careful to look for warnings that say contains phenylalanine in order to make sure that they don't have too much of this. Additionally, albinism, so being an albino, is because the enzyme that converts tyrosine to melanin is defective. So melanin comes from tyrosine. So their body literally just doesn't have the enzyme to make melanin. And that's why their skin is all white. So here's a table of genetic diseases. I'm not going to go through these specifically, but if you're interested, you can see what different changes occur in the genetics for these genetic diseases. So real quick, our one little practice problem is going to be identify each type of mutation as a point deletion or insertion mutation. So cysteine enters the DNA sequence, meaning it's added in. This would be an insertion. When adenosine is removed from a DNA sequence, so completely pulling it out, is a deletion. Lastly, a base sequence of TGA and DNA changes to TAA. So we're just switching one for the other. There's no frame shift. So this would be a point mutation.